Good evening and welcome to Dreamy Academy. Tonight we embark on a journey into the intricate and wondrous world of life itself, exploring the very fundamentals of zoology. As we begin, I'd love to know where you are joining me from, your city, your country, perhaps even what the night sky looks like outside your window. If you find peace and fascination in these quiet explorations of science, please consider taking a moment to like this video and subscribe. It truly helps this tranquil corner of curiosity to flourish. And to those already part of our thoughtful community, thank you sincerely for your presence and support. Tonight, we will gently drift into a question so fundamental it often escapes our notice. What does it truly mean to be an animal? It's a query that seems simple, yet its answer unfolds into a tapestry of deep time, elegant chemistry, and astonishing adaptation. Imagine for a moment the incredible lineage you carry within you. Every cell in your body, every beat of your heart is part of a story that stretches back billions of years, connecting you to the very first stirrings of multicelled life. You're not merely an observer of this grand narrative. You are a living, breathing chapter of it. Zoology, at its heart, is the study of this magnificent animal kingdom, a realm of creatures that move, that hunt, that build, that sing, that burrow, that soar. It's a field that rewards patient contemplation, revealing not just a collection of facts, but a profound interconnectedness, a shared heritage woven through the intricate dance of survival and flourishing. It is the story of how matter learned to be alive, to respond, to adapt, and to dream. So let us begin our quiet exploration, allowing the beauty and depth of this understanding to gently lull us into a peaceful wonder. Have you ever paused to truly consider the ordinary miracle of a housefly buzzing past your ear, or the quiet patience of a spider weaving its web? What is it precisely that makes these creatures, and indeed ourselves, so fundamentally different from the oak tree outside your window, or the moss clinging to a damp stone? It is a question that leads us to the very bedrock of biology, to the defining characteristics that whisper, "This is an animal." Let's begin by considering the most fundamental distinctions. The world around us is teeming with life, yet within that vastness, there are distinct ways of being alive. Plants, rooted and still, draw their energy directly from the sun, converting light into life. Fungi, like silent alchemists, break down the world around them, recycling nutrients in the soil's dark embrace. But animals, we are different. We are the movers, the eaters, the responders. The first defining whisper of an animal is its multicellularity. Unlike the single-celled bacteria that teem in every drop of water, or the microscopic protists that dance unseen, animals are composed of many cells working in concert. Imagine each cell as a tiny, specialized worker in a grand living city. Some cells form the strong supporting walls of bone, others the intricate wiring of nerves, still others the soft protective layers of skin. This division of labor, this elegant cooperation among countless cells, allows for a complexity far beyond what a single cell can achieve. It means that an animal can grow large, develop intricate structures, and perform a dazzling array of functions, all because its cells have learned to specialize and collaborate, each sacrificing a measure of independence for the greater good of the whole. The second whisper is heterotrophy. This simply means that animals must consume other organisms to obtain their energy and the building blocks for their own bodies. Unlike plants, which are autotrophs, self-feeders creating their own food from sunlight, animals cannot simply photosynthesize. We must eat. This fundamental need to acquire sustenance has shaped every aspect of animal life, from the sharp teeth of a predator to the filtering mouthparts of a baleen whale, from the swift flight of a bird chasing an insect to the slow, deliberate crawl of a snail grazing on algae. The entire intricate web of life, the delicate balance of predator and prey, herbivore and plant, is built upon this essential characteristic of animals seeking out and consuming others. And this leads us to the third, often most visible whisper: movement. While not every animal moves in the obvious ways we might think of, some, like sponges, remain fixed to a spot. The capacity for active locomotion is a hallmark of the animal kingdom. The need to find food, to escape predators, to seek mates, to explore new territories, has driven the evolution of an astonishing array of appendages, muscles and nervous system. From the rhythmic undulations of a jellyfish propelling itself through the ocean, to the powerful leap of a deer, to the precise manipulation of tools by a primate, movement is the answer to the heterotrophic imperative. It is the physical expression of a creature's will to survive and thrive in a dynamic world. Consider for a moment the simplest of these multicellular animals, the sponges. These ancient creatures, appearing in the fossil record hundreds of millions of years ago, offer us a glimpse into the very dawn of animal life. A sponge is essentially a porous bag of cells, each performing a relatively simple task. There are cells that create currents to draw water in, cells that filter tiny food particles from that water, and cells that provide structural support. They lack true tissues, organs, or even a nervous system. Yet they are undeniably animals, multicellular and heterotrophic, quietly filtering vast quantities of water, a living testament to the power of cellular cooperation. From these humble beginnings, a greater complexity arose, giving rise to animals with a more organized body plan, like the stolnidarians, a group that includes jellyfish, sea anemones, and corals. Here we see the emergence of radial symmetry, where the body parts are arranged around a central axis, like the spokes of a wheel. A jellyfish drifting through the water can respond to stimuli coming from any direction. Its tentacles, armed with stinging cells, are equally ready to capture prey or defend against a threat, no matter from which angle it approaches. This simple, elegant symmetry is perfectly suited for a life suspended in water, where dangers and opportunities can arise from any direction. The evolution of bilateral symmetry, where an animal has a distinct left and right side, a front and a back, and a top and a bottom, marked another profound shift. Imagine a flatworm creeping along the seafloor. It has a head with sensory organs concentrated there, allowing it to move forward into new environments, sensing what lies ahead. This kind of symmetry is intimately linked to active, directed movement, allowing for more efficient hunting, burrowing, and escaping. The vast majority of animals, from insects to fish to humans, exhibit bilateral symmetry, a testament to its evolutionary success in navigating a complex world. All these diverse forms, from the simplest sponge to the most complex mammal, share a common thread. They are built from a foundational blueprint. They hint at a shared ancestry, a deep branching tree of life where every creature, no matter how different, is connected by these fundamental biological principles. The very existence of such a vast and varied animal kingdom, all united by these core characteristics, is a testament to the elegant solutions life has found to the challenges of existence. It inspires a profound humility, a sense of belonging to this grand unfolding story. So we've seen how animals are defined by their multicellular nature, their need to consume others for energy, and their capacity for movement, all built upon foundational body plans like radial or bilateral symmetry. But these definitions are just the beginning. How does this basic blueprint, this shared heritage, allow for such an astounding array of forms, functions, and behaviors? How does life itself, across countless generations, adapt and change to fill every conceivable niche on our planet? That question leads us into the quiet, patient engine of transformation that has shaped every living thing, the process of evolution, which we will explore in our next quiet hour together. Welcome back, dear friends, to our tranquil corner of curiosity here at Dreamy Academy. As we gently drift further into the intricate and wondrous world of zoology, we carry with us the foundational understanding from our last gathering, the shared blueprint that defines an animal. We explored the elegant simplicity of multicellularity, the vital necessity of heterotrophy, and the profound capacity for movement, noting how these principles sculpt even the most ancient and seemingly simple forms of life. Now, I invite you to pause with me for a moment to truly consider the magnificent panorama that stretches out from this shared origin. If all animals begin with a similar biological essence, a common ancestor whispering promises of life in the deep past, how then do we account for the breathtaking, bewildering diversity that surrounds us? How does a microscopic tardigrade, an armoured marvel that defies the very notion of fragility, share a common heritage with the colossal blue whale, a gentle leviathan that navigates the vast ocean depths? How does the silent, patient starfish rooted to the seabed connect to the swift, soaring peregrine falcon, a feathered arrow across the sky? This, my thoughtful companions, is where the grand narrative of evolution unfurls itself. It is the story of change, of adaptation, of life's ceaseless dance with the environment across unimaginable stretches of deep time. Evolution is not a destination but a journey, a continuous flowing process that sculpts, refines, and diversifies all living things, including every single animal we encounter. Imagine, if you will, a single mighty river, its source high in the mountains. As it cascades downward, it branches and meanders, carving new paths, shaping the landscape, each tributary developing its own unique character, while remaining fundamentally connected to the original flow. This, in essence, is the story of life, ever branching, ever diversifying, yet eternally intertwined. At the heart of this transformative process lies a principle of profound elegance and simplicity: natural selection. Have you ever considered how perfectly suited each creature seems to be for its particular niche in the world? The chameleon's camouflage, the eagle's keen eyesight, the bat's echolocation, these are not random occurrences, but the semzit results of countless generations of subtle refinement. Natural selection in its essence is the differential survival and reproduction of individuals due to differences in phenotype. But let us break that down gently into more accessible terms. Consider any population of animals, perhaps a group of deer in a forest or a school of fish in the ocean. Within any such group we observe natural variation. No two individuals are ever precisely identical. Some are slightly taller, some quicker, some possess a slightly different coloration or a subtly more efficient metabolism. These variations, often minute, are the raw material upon which evolution acts. Now introduce the undeniable reality of life. Resources are finite. Food, shelter, mates, these are not limitless. This leads, inevitably, to a gentle, yet persistent struggle for existence. Not a violent, dramatic battle necessarily, but a constant negotiation with the environment. In this delicate balance, individuals whose inherited traits make them slightly better equipped to survive and reproduce in their specific environment are more likely to pass on those advantageous traits to the next generation. Perhaps a deer with slightly longer legs can outrun a predator more effectively, or a fish with a subtler pattern can evade detection. Over vast spans of time, across innumerable generations, these small incremental advantages accumulate. Those traits that confer a benefit become more common in the population, while those that are less advantageous gradually diminish. This is not a conscious striving on the part of the animal, but rather an unconscious, relentless shaping by the forces of nature. It is a slow, patient sculptor, chiseling away at the rough stone of variation, revealing the refined form beneath. We often hear the phrase survival of the fittest, and it's worth pausing to understand what fittest truly means in this context. It does not necessarily mean the strongest or the fastest, but rather the most suited or adapted to a particular set of environmental conditions. The fittest are those who leave the most viable offspring, ensuring their genetic legacy continues. But from where do these precious variations arise? What are the silent alchemists that continuously provide the raw material for natural selection to work its magic? Here, we turn our gaze to the microscopic world within each cell, to the very bedrock of heredity, genetics. The primary source of new variation is mutation. Mutations are spontaneous, random changes in an organism's DNA sequence. Imagine a grand ancient manuscript, meticulously copied over millennia. Occasionally a scribe might make a tiny error. A single letter changed, a word subtly altered. Most of these changes might be neutral, having no discernible effect. Some might be detrimental, making the text less coherent. But on rare occasions, a change might actually improve a passage, adding clarity or new meaning. In the biological world, these errors can lead to new traits, a slight alteration in a protein, a minor shift in a developmental pathway. These are the sparks of novelty, the random twists of fate that introduce new possibilities into the genetic pool. Another powerful engine of variation, particularly in sexually reproducing animals, is the recombination of genetic material. With each new generation, the genetic instructions from two parents are shuffled and combined in unique ways, creating an entirely new genetic hand for the offspring. This constant reshuffling ensures that even without new mutations, every individual is a unique mosaic of traits, a fresh combination of ancestral legacies. Together, mutation and recombination ensure that the genetic tapestry of a population is never static, always shimmering with possibilities, always providing the grist for natural selection's mill. The beautiful outcome of this interplay between variation and selection is adaptation. An adaptation, simply put, is any heritable trait that helps an organism survive and reproduce in its current environment. It is the perfect fit of a key into a lock, the precise alignment of form and function with ecological demands. Consider the elongated neck of a giraffe, an adaptation that allows it to browse on leaves high above the reach of other herbivores, or the streamlined body of a dolphin, exquisitely adapted for swift movement through water. These are not traits that appeared overnight, but rather the culmination of countless generations, each tiny advantage building upon the last, until the animal becomes a living testament to its environment. Adaptations are dynamic. As environments change, so too does the selective pressure, leading to new adaptations or the modification of existing ones. It is a continuous conversation between life and its surroundings, and what happens when these adaptations accumulate to such an extent that populations diverge, profoundly they can no longer interbreed. This, my dear friends, is the process of speciation, the birth of new species, Imagine our single river again, branching into two distinct streams. Over vast geological timescales, perhaps a physical barrier like a mountain range or a new ocean current separates a population. Isolated from one another, these two groups experience different selective pressures, accumulating different mutations and adaptations. What might be advantageous in one environment could be detrimental in another. Gradually, the genetic differences between them become so pronounced that even if the barrier were removed, they would no longer recognize each other as potential mates, or if they did, their offspring would be infertile. They have become distinct species, two new branches on the tree of life, each destined to follow its own unique evolutionary path. This journey through evolution, from the subtle shifts of mutation to the grand branching of speciation, unfolds not in moments or even centuries, but across the awe-inspiring canvas of deep time. We speak of millions, even billions of years, a scale that can humble the human mind. Each fossil we unearth, each genetic similarity we discover, is a whispered testament to this ancient ongoing story. It is the story of resilience, of innovation, of an unbroken chain of life stretching back to the very first cells. And every animal, from the simplest sponge to the most complex primate, carries within its very being the echoes of this journey, a testament to shared ancestry and the relentless creative power of evolution. As we contemplate this grand tapestry woven through deep time, we are reminded that despite the incredible diversity, all animals are fundamentally connected. They are all expressions of the same biological principles, shaped by the same evolutionary forces. This understanding provides a profound sense of unity, a shared heritage that binds us all within the magnificent animal kingdom. In our next quiet exploration, we will begin to navigate this astonishing diversity. How do we make sense of such an overwhelming array of forms and functions? How do we categorize and understand the relationships between these myriad creatures, all born from the same evolutionary processes? We will begin to explore the elegant systems of classification that allow us to map the branching pathways of life, placing each animal within its proper context on the grand tree of existence. Until then, I encourage you to carry this sense of wonder with you, to see the whispers of evolution in every living creature you encounter, from the smallest insect to the largest mammal. The story, as you now know, is truly magnificent. Welcome back, dear friends, to our tranquil corner of curiosity here at Dreamy Academy. We've journeyed through the fundamental characteristics that define animals, explored the astonishing tapestry of deep time woven by evolution, and witnessed how the silent, relentless currents of natural selection sculpt the myriad forms we see around us. We've acknowledged the profound truth that despite their dizzying diversity, all animals share a common ancient lineage, a single branch on the grand tree of life. Now, as we stand at the precipice of this immense biological richness, a new question gently rises within us, doesn't it? How do we even begin to navigate such an intricate and wondrous world? How do we bring order to this magnificent, sprawling chaos of life, to understand its relationships and its history without being overwhelmed by its sheer scale? Imagine for a moment an impossibly vast library, its shelves stretching beyond the horizon, filled with every story ever told, every whisper of life ever uttered. Without a system, without a catalogue, how would we ever find the tales that speak to us most deeply, or understand how they connect? This, in essence, is the challenge that zoologists, indeed all biologists, have sought to address for centuries. And the answer we find lies in the elegant, ever-evolving science of taxonomy and systematics. these are the guiding stars that help us map the constellations of life, allowing us to group, name and classify organisms based on their shared characteristics and, crucially, their evolutionary heritage. Have you ever paused to truly consider the names we give to the creatures that share our planet? A lion, a house cat, a tiger. We instinctively recognize their kinship, yet understand their distinct identities. This isn't merely an arbitrary labeling exercise. It's a profound attempt to reflect the very architecture of life. For a long time, early naturalists grappled with this, often relying on superficial resemblances or local names, which, while charming, lack the universality and precision needed for a global understanding. Then, in the 18th century, a Swedish botanist named Carolus Linnaeus gifted us a system that, in its essence, we still use today. Linnaeus, with his meticulous eye and groundbreaking insights, introduced what we know as binomial nomenclature, the two-part naming system for every species. You've encountered it countless times, perhaps without even realizing its profound significance. Homo sapiens, Panthera leo, Canis familiaris. These are not just names, but concise, universally understood labels that immediately place an organism within its broader biological family. The first part, the genospore, groups closely related species, while the second part, the specific epithet, distinguishes that particular species within its genus. Together, they form a unique scientific name, a passport for that species recognized by scientists across the globe, transcending language barriers and local colloquialisms. It's a testament to Linnaeus's genius that this system, born centuries ago, remains the bedrock of how we communicate about life's diversity. But Linnaeus's vision extended beyond just naming. He proposed a hierarchical system of classification, a nested series of categories that would group organisms into increasingly inclusive sets, much like organizing files in folders within larger folders. This structure, often referred to as the Linnaean hierarchy, provides a framework for understanding relationships from the most specific to the most general. Let us gently drift through these levels, contemplating what each signifies. At the very top, encompassing all life, we have the domain. This is the broadest category, dividing all organisms into three fundamental groups: bacteria, archaea, and eukarya. Since animals like plants, fungi, and protists are characterized by complex cells with a nucleus and other membrane-bound organelles, we, along with all the animals we will explore, belong to the domain eukarya. Think of the domain as the entire library building itself, housing all the books ever written. Stepping down, we arrive at the one kingdom. Within the domain eukarya, we find several kingdoms, and our focus, of course, is the kingdom Animalia. This is where we gather all the multicellular, heterotrophic, actively moving organisms we discussed in Chapter 1. If the domain is the entire library, then the kingdom is a major wing or floor dedicated to a particular type of literature, say, all works of fiction. Below the kingdom, we encounter the phylum, plural, phyla. This level groups organisms based on fundamental body plans and organizational features. For instance, the phylum Chordata includes all animals with a notochord, a dorsal hollow nerve cord, pharyngeal slits, and a postanal tail at some point in their development, a group that includes fish, amphibians, reptiles, birds, and mammals. The phylum Arthropoda, on the other hand, encompasses insects, spiders, and crustaceans, all characterized by an exoskeleton and jointed appendages. Imagine the kingdom's fiction wing now divided into genres, perhaps a section for epic fantasy, another for historical novels, and so on. Each phylum represents a distinct ancient lineage with a unique blueprint for life. Our journey continues to the class, or within each phylum, organisms are further refined into classes based on more specific shared characteristics. Within the phylum Chordata, for example, we find the class Mammalia, mammals, characterized by mammary glands, hair, and a four-chambered heart. The class Aves, birds, with feathers and hollow bones, or the class Reptilia, reptiles, with scales and ectothermy. These are like subgenres within our library. Perhaps within epic fantasy, we now have subsections for high fantasy, urban fantasy, or dark fantasy. Next is the order. This level groups related families. For instance, within the class Mammalia, the order Primates includes monkeys, apes, and humans, sharing characteristics like grasping hands and feet, large brains, and binocular vision. The order Carnivora, another mammalian order, comprises animals like cats, dogs, bears, and seals, all adapted for a meat-eating diet. Continuing our library analogy, the order might be a shelf or a collection of books by a particular school of authors within a subgenre. Moving closer to the individual species, we have the family. This category groups closely related genera. For example, within the order Carnivora, the family Felidae includes all cats, lions, tigers, domestic cats, while the family Canidae includes all dogs, wolves, foxes, domestic dogs. These are distinct yet clearly related groups. In our library, this would be a specific series of books by a particular author, or perhaps a collection of works by authors known for a very similar style. Finally, we arrive at the genus, plural, genera, and the whole species. As we discussed with binomial nomenclature, the genus groups very closely related species that share a recent common ancestor. Panthera, for example, is the genus that includes lions, Panthera leo, tigers, Panthera tigris, leopards, Panthera pardus, and jaguars, Panthera onca, and then the species itself, the fundamental unit of classification, representing a group of organisms that can interbreed and produce fertile offspring. This is the individual book, unique and complete in itself, yet part of a larger literary tradition. This hierarchical system, from domain down to species, is not merely a convenient way to organize information. It is a profound reflection of evolutionary relationships. When Linnaeus first devised his system, he based his classifications on visible shared characteristics. He could not have known the invisible threads of DNA that connect all life, nor the grand narrative of descent with modification. Yet remarkably, his framework often aligns beautifully with what modern genetic analysis reveals about phylogeny, the evolutionary history and relationships among groups of organisms. Today, systematics goes beyond just physical traits. With the advent of molecular biology, we can compare DNA sequences, providing an incredibly precise lens through which to view evolutionary kinship. These genetic blueprints allow us to trace lineages back through deep time, confirming some of Linnaeus's groupings and, at times, prompting us to redraw parts of the tree of life. This means that our understanding of classification is not static. It's a dynamic, living science, constantly being refined and updated as new discoveries emerge from fossil records, genetic labs, and field expeditions. It's a testament to the scientific process itself, a continuous journey of discovery, questioning, and refinement, always striving for a clearer, more accurate picture of reality. So, as we prepare to embark on our quiet explorations of specific animal groups, remember this intricate scaffolding of classification. It's the language we use, the map we follow, to understand how the incredible diversity of animal life has unfolded from shared ancestors. It helps us see not just individual species, but the deep interconnected web of life, revealing the common threads that bind a microscopic tardigrade to a colossal blue whale, a delicate butterfly to a powerful eagle. In our next quiet contemplation, we will begin to delve into the very first branches of the animal kingdom, gently turning the pages of life's oldest stories. We'll start our journey with some of the simplest yet utterly fascinating animal forms, those that laid the foundational blueprints for all the complexity that followed. We'll marvel at their adaptations, their unique strategies for survival, and their enduring presence in the world, reminding us that every creature, no matter how humble, holds a vital place in the grand narrative of life. Until then, I encourage you to look around you, perhaps even at the creatures just outside your window, and ponder the vast, unseen hierarchy that connects them all. What an astonishing world we share, isn't it? As we gently drift from the intricate maps of taxonomy and the grand narratives of evolutionary history, our journey now calls us to a more tangible exploration. Imagine, if you will, stepping onto the very bedrock of animal life, a place where the foundational principles we've so carefully laid out begin to manifest in forms both ancient and profoundly simple, yet utterly astonishing. We have charted the course, we've understood the language, and now, dear friends, we prepare to meet our first inhabitants of this wondrous kingdom. Our inaugural destination takes us beneath the shimmering surface of the world's waters, into realms both sunlit and shadowed, where life often unfolds with a quiet, persistent elegance. Here, we encounter the phylum Porifera, the sponges. Have you ever paused to truly consider these unassuming beings? Often mistaken for plants or mere geological formations, sponges are in fact quintessential animals, embodying the very first whispers of multicellularity, heterotrophy, and movement, albeit in their most primordial expressions. Let us return to our core definition from Chapter 1, shall we? We established that animals are multicellular, heterotrophic, and capable of movement at some stage of their life. The sponges, in their elegant simplicity, beautifully illustrate these tenets, inviting us to witness their unfolding in a manner that truly grounds our understanding. First, consider their multicellularity. Unlike the complex tissues and organs we associate with more familiar animals, sponges present a more loosely organized cellular structure. Imagine a bustling cooperative community where specialized cells work together, yet retain a remarkable degree of independence. There are no true tissues here, no nervous system to coordinate grand movements, no digestive organs in the conventional sense. Instead, we find a fascinating array of cell types, each performing its specific role, much like tiny specialized workers in a grand living city. This modularity is a testament to their ancient lineage, offering us a glimpse into the very dawn of animal complexity. It's as if evolution, in its early experiments, was testing the waters of cooperation, finding success even in this seemingly unorchestrated harmony. Then we turn to heterotrophy, the fundamental need to consume other organisms for energy. Sponges are masters of this, yet their method is uniquely their own, a marvel of biological engineering known as filter feeding. Picture a living pump tirelessly drawing vast quantities of water through its body. This is the work of specialized cells called choanocytes, or collar cells. Each choanocyte possesses a flagellum, a whip-like appendage that beats rhythmically, creating a gentle current. Encircling the base of this flagellum is a delicate collar of microvilli, like a microscopic sieve. As water is drawn in through countless tiny pores, or ostia, Across the sponge's outer surface, these choanocytes capture microscopic food particles, bacteria, plankton, detritus, from the passing current. It is a continuous silent feast, a testament to efficiency and adaptation. Nutrients are then distributed throughout the sponge by another type of cell, the pure amoebocytes, which crawl through the jelly-like matrix within the sponge, delivering sustenance and carrying away waste. Truly a scent alchemist, transforming the invisible into the tangible. And finally see movement. Here, the sponges offer us a nuanced perspective. While adult sponges are largely sessile, meaning they remain fixed to a substrate for their entire lives, their larval forms tell a different story. Many sponge species produce free-swimming larvae, minute motile entities that drift through the water column, propelled by cilia. These tiny travellers are crucial for dispersal, allowing sponges to colonize new areas and expand their populations. So while the mature sponge may appear as static as a stone, the capacity for movement, a hallmark of animal life, is undeniably present within its life cycle. A fleeting dance of discovery before settling into a lifetime of quiet contribution. It reminds us that movement can take many forms and serve various purposes throughout an animal's existence. Let us delve a little deeper into the elegant architecture of these quiet beings. A sponge's body plan, while lacking true symmetry in many cases, though some exhibit a rudimentary radial symmetry, is fundamentally built around this filter-feeding mechanism. Water enters through the ostia, flows into a central cavity known as the spongocoel, and then exits through a larger opening called the osculum. Imagine a living vessel, intricately porous, designed solely for the gentle flow of water. To maintain this structure, sponges rely on an internal skeleton. This skeletal framework can be composed of needle-like spicules, made of either calcium carbonate or silica, which provide rigidity and protection. Other sponges, like the familiar bath sponge, are supported by a softer, fibrous protein called spongin. These diverse skeletal elements, often beautiful under a microscope, are a testament to the varied solutions evolution has devised for structural integrity. The resilience and adaptability of sponges extend to their methods of reproduction. They are remarkably versatile. Asexually, they can reproduce through budding, where a small new sponge simply grows out from the parent, through fragmentation, where a broken piece can regenerate into a whole new individual, and even through the formation of gemmules, dormant, tough-coated buds that can survive harsh conditions and later develop into new sponges. Sexually, most sponges are hermaphroditic, possessing both male and female reproductive cells. They typically release sperm into the water, which are then drawn into another sponge, where fertilization occurs internally. The resulting larvae are then released to embark on their motile journey before settling down. This combination of reproductive strategies ensures their continued presence across diverse marine and freshwater environments, a quiet triumph of biological persistence. And what of their role in the grand tapestry of life? Sponges, despite their unassuming nature, are vital members of aquatic ecosystems. They are nature's silent purifiers, filtering immense volumes of water daily, improving water quality by removing suspended particles and microorganisms. This constant filtering contributes significantly to the clarity and health of their environments. Furthermore, their intricate structures provide crucial habitat and shelter for countless small invertebrates, fish, and other marine life, creating microecosystems within their own bodies. They are also a food source for a select few specialized predators, such as certain sea slugs and turtles, demonstrating that even in their simplicity, they are woven into the complex food webs of the ocean. As we contemplate the sponges, we are not merely observing a fascinating animal group, we are peering back into the annals of deep time. Sponges represent one of the earliest diverging animal lineages, their fossil record stretching back hundreds of millions of years, offering us a living connection to the very first multicellular animals that graced our planet. They are, in a sense, living fossils, carrying within their cellular organization echoes of a time before true tissues, before complex organs, before the intricate symphonies of nervous systems. Their continued success, their quiet persistence across geological eras, is a profound testament to the power of fundamental biological principles. So as you reflect upon the humble sponge, perhaps next time you encounter an image or description of one, you will see beyond its static form. You will perceive the tireless filter feeder, the ancient multicellular pioneer, the quiet architect of clean waters, and the subtle embodiment of animal characteristics in their most foundational expression. We began our zoological journey by defining what it means to be an animal, and in the sponges, we find those definitions etched into the very fabric of their ancient, resilient lives. This quiet exploration of Porifera has offered us a vital first step, a gentle immersion into the astounding diversity that awaits us. From these loosely organized cellular communities, we will soon begin to trace the evolutionary pathways that led to increasingly complex forms, to the emergence of true tissues, and to the myriad wonders that populate the animal kingdom. But for now, let us savor the profound lessons offered by these quiet architects of the deep. Their story is a powerful reminder that even the simplest forms of life hold boundless wisdom and beauty, inviting us to a deeper appreciation of every living thing. We carry their ancient whispers with us as we prepare to venture further into the next chapter of this grand narrative of life. Welcome back, dear friends, to our tranquil corner of curiosity at Dreamy Academy. We've journeyed through the foundational strata of animal life, marveling at the eloquent simplicity and profound evolutionary significance of the sponges, those storied ancient architects of the seabed. We saw how their cells, those specialized and cooperative, remained largely independent, forming a community rather than a truly integrated whole. We observed their unique filter-feeding prowess, their unassuming yet vital role in purifying the oceans, and their quiet testament to life's earliest multicellular stirrings. Now, as we gently drift forward in the grand narrative of life, we arrive at a truly pivotal moment, a leap in biological organization that forever changed the course of animal evolution. Imagine, if you will, the tapestry of deep time, where the threads of life are continually rewoven, each new pattern building upon the last. From the elegant, porous architecture of the sponges, we now turn our gaze to a group of animals that took the concept of cellular cooperation to an entirely new level, the development of true tissues. Have you ever paused to truly consider what a tissue truly means in the biological sense? It's more than just a collection of cells, it's a group of similar cells working together to perform a specific function, bound by common purpose and often by extracellular material. This organized collaboration allows for a division of labor that is far more efficient and complex than what we observed in the sponges. This is the hallmark of our next fascinating phylum, Cnidaria. When we speak of Cnidaria, we're inviting you to explore a world of breathtaking beauty and startling effectiveness. Think of the mesmerizing dance of a jellyfish, the vibrant stony cities built by corals, or the delicate swaying grace of a sea anemone. These are the cnidarians, and they represent a profound evolutionary advancement, distinguished by the presence of two distinct embryonic germ layers, the ectoderm and the endoderm, that develop into true tissues. This makes them diploblastic, a term that elegantly describes their two-layered body plan, setting them apart from the simpler sponges and paving the way for the more complex triploblastic animals we will encounter later. As you contemplate these creatures, we invite you to appreciate their fundamental body plan, a sac-like structure with a single opening that functions as both mouth and anus. This opening leads into a central gastrovascular cavity, where digestion and nutrient distribution take place. It's a remarkably efficient design for animals that capture their food from the surrounding water, allowing for both ingestion and egestion through the same aperture. But perhaps the most iconic and defining characteristic of the cnidaria, the very feature that gives them their name, is the presence of specialized stinging cells called cnidocytes. These microscopic marvels are veritable biological harpoons, each containing a barbed, venom-filled capsule called a dermatocyst. Imagine for a moment a tiny spring-loaded weapon coiled and ready within each cell. When triggered by touch or chemical cues, often from unsuspecting prey, the nematocyst rapidly discharges, injecting venom or entangling the victim. It is this astonishing innovation that allows cnidarians, many of whom are sessile or slow-moving, to effectively capture prey and defend themselves. It's a testament to the ingenious solutions wrought by natural selection, a silent whisper of life's endless creativity. Another significant leap forward for the cnidarians is the emergence of a rudimentary nervous system. Unlike the sponges, which communicate cell to cell without a centralized system, cnidarians possess a nerve net. This diffuse network of interconnected neurons spreads throughout their body, allowing them to sense their environment and coordinate simple responses, such as muscle contractions for movement or the coordinated firing of cnidocytes. While not a centralized brain, this nerve net represents the very beginnings of sensory processing and coordinated action in the animal kingdom, a truly foundational step in the evolution of behavior. As we delve deeper into the cnidaria, we discover a remarkable duality in their body forms, often cycling between two distinct stages, the raw polyp and the medusa. The polyp form is typically sessile, meaning it is attached to a substrate. Picture a sea anemone rooted to the ocean floor, its tentacles swaying gently in the currents, mouth facing upwards. Or consider the individual polyps that make up a coral colony, each a tiny, soft-bodied animal secreting its stony skeleton. These polyps are generally cylindrical, with a mouth surrounded by tentacles at one end and a base attached to a surface at the other. In contrast, the medusa form is free-swimming, bell-shaped, and often epitomizes what we commonly call a jellyfish. With its mouth typically facing downwards and its tentacles trailing below, the medusa propels itself through the water by rhythmic contractions of its bell, a mesmerizing ballet of propulsion. Many cnidarians exhibit a life cycle that alternates between these two forms, with polyps often reproducing asexually and budding off medusae, which then reproduce sexually, releasing larvae that settle to form new polyps. It's a sophisticated reproductive strategy that allows them to exploit different ecological niches throughout their lives. Let us now cast our gaze across the astonishing diversity within the cnidaria, revealing the myriad ways this basic body plan has been adapted and refined over millions of years. we encounter the hydrozoa, a class that includes both solitary polyps like the freshwater hydra, which can regenerate itself with incredible tenacity, and colonial forms like the infamous Portuguese man-o'-war. Though often mistaken for a single organism, the man-o'-war is a colony of specialized polyps and medusae, each performing a specific function, some for feeding, some for reproduction, some for defense with extraordinarily long and potent stinging tentacles. It's a breathtaking example of superorganismal complexity built from simpler units. Then there are the cyphozoa, the true jellyfish. These are the creatures that inspire a sense of ethereal wonder and sometimes a healthy respect for their stinging power. Their large pulsating bells, often translucent and adorned with intricate patterns, glide through the water, their long trailing tentacles and oral arms sweeping for prey. To watch a jellyfish drift is to witness a silent poem of motion, a testament to the elegance of radial symmetry in action. Perhaps the most familiar and ecologically significant cnidarians belong to the class Anthozoa, which includes all sea anemones and corals. Sea anemones, with their vibrant colours and flower-like appearance, are essentially large solitary polyps, often forming symbiotic relationships with other creatures like the clownfish, which finds refuge among their stinging tentacles. Corals, however, are the true architects of the marine world. These tiny polyps, living in vast colonies, secrete calcium carbonate skeletons, building immense structures over millennia that form the backbone of coral reefs. Imagine for a moment the sheer scale and complexity of a coral reef. It is a bustling underwater metropolis, a vibrant ecosystem teeming with an unparalleled diversity of life. These reefs, built by countless generations of tiny coral polyps, provide food, shelter, and breeding grounds for a quarter of all marine species, despite covering less than 1% of the ocean floor. They protect coastlines from erosion, cycle nutrients, and contribute significantly to the oxygen we breathe. The silent alchemists of the Anthozoa, through their patient, collective work, create environments that pulse with life, a profound example of how seemingly simple organisms can have an immeasurable impact on the planet. And finally, a brief mention of the Cubozoa, or box jellyfish. These fascinating, though often dangerous canarians possess a more advanced nervous system than other jellyfish, including true eyes with lenses, allowing them to navigate and hunt with surprising agility. Their potent venom makes them creatures of both scientific interest and cautionary tales. As we conclude our exploration of the Cnidaria, we hope you feel a renewed sense of awe for these remarkable animals. They stand as a testament to a crucial turning point in evolutionary history, the emergence of true tissues, a functional gastrovascular cavity, a rudimentary nervous system, and the ingenious cnidocytes. From the simple cooperative cells of sponges, we have now witnessed the development of organized, specialized tissues, laying the groundwork for the more complex organ systems that will define the animal kingdom's subsequent diversification. We have moved beyond the realm of cellular aggregates into the world of integrated body plans, where cells work in concert within tissues to achieve greater efficiency and more complex behaviors. This fundamental step opened up vast new possibilities for animal life, enabling more sophisticated interactions with the environment and paving the way for the incredible array of forms and functions we see in the animal kingdom today. In our next chapter, we will continue this journey, venturing into the fascinating world of the bilaterally symmetrical animals, those creatures that possess a distinct head and tail, a left and right side. We will explore the evolutionary advantages this new body plan conferred and meet the earliest representatives of this incredibly diverse lineage. Until then, we encourage you to carry with you the image of the mesmerizing jellyfish, the resilient anemone, and the world-building coral, appreciating the profound elegance and evolutionary significance of the cnidaria. Their whispers of life continue to resonate through the silent depths, reminding us of the enduring wonder that is zoology. And so, with our quiet explorations, we've journeyed from the foundational simplicity of sponges, those living filters of the aquatic realm, to the elegant radial symmetry and nascent tissues of the cnidarians. We observed the delicate dance of polyps and medusae, marveling at the innovation of stinging nematocysts and the first whispers of a nervous system. It was a profound evolutionary leap, wasn't it? The emergence of true tissues, born from those two primordial germ layers, marked a pivotal moment in life's grand narrative, allowing for greater specialization and complexity. But as we gently drift through the currents of evolutionary time, a new blueprint begins to emerge, promising even more intricate designs and astonishing adaptations. Imagine, for a moment, a world where creatures no longer simply sense their environment from all directions, but rather move with purpose, with a clear front and back, a distinct top and bottom. Have you ever paused to truly consider the subtle yet profound shift this would entail? This brings us to one of the most significant architectural innovations in the animal kingdom: bilateral symmetry. If you were to draw an imaginary line down the center of your own body from head to toe, you would find that your left half is, for the most part, a mirror image of your right half. This is bilateral symmetry, a design that has proven incredibly successful, giving rise to the vast majority of animals we encounter today, from the smallest insects to the largest whales, and indeed to ourselves. What, you might wonder, is so revolutionary about this particular arrangement? Its advantages are manifold and deeply interconnected. Firstly, bilateral symmetry inherently leads to bicephalization, a term we use to describe the concentration of sensory organs and nerve tissue, a brain in essence, at the anterior or front end of the organism. Think about it. If you're moving forward through an environment, it makes perfect sense to have your eyes, nose, and ears leading the way, gathering information about what lies ahead. This allows for far more direct and efficient movement, enabling animals to actively seek out food, mates, or escape from predators, rather than passively waiting for the world to come to them. It's the difference between a stationary anemone waving its tentacles indiscriminately and a swift predator tracking its prey. Secondly, this distinct anterior-posterior head-to-tail axis, coupled with a dorsal-ventral back-to-belly axis, allows for specialized regions of the body to develop. The front can be dedicated to sensing and feeding, the middle to digestion and locomotion, and the rear to excretion and reproduction. This division of labor within the body itself is a hallmark of complexity, laying the groundwork for the sophisticated organ systems we'll soon explore. But bilateral symmetry is not the only architectural marvel we encounter at this juncture. Hand in hand with this new body plan comes another profound developmental innovation: triploblasty. Remember how cnidarians developed from two embryonic germ layers, the ectoderm and the endoderm, making them diploblastic? Well, with the emergence of bilaterally symmetrical animals, a third germ layer arises, the mesoderm. This mesoderm is, in many ways, the great sculptor of internal complexity. Nestled between the outer ectoderm and the inner endoderm, this middle layer gives rise to a wealth of new structures that were simply not possible before. Think of muscles, which allow for powerful and controlled movement, the intricate components of the circulatory system, transporting nutrients and oxygen throughout the body, the skeletal system providing support, and many of the sophisticated organs that will define later animal groups. The mesoderm is the raw material for internal scaffolding for the machinery of motion and for the vessels that connect it all. It's a testament to the quiet ingenuity of evolution, adding a whole new dimension to the building blocks of life. And so, as we cross this threshold into the realm of bilateral symmetry and triploblasty, we encounter our next fascinating chapter in zoology, the phylum Platyhelminthes, more commonly known as the flatworms. These unassuming creatures, often overlooked in their quiet corners of the world, are profound pioneers. They represent some of the earliest animals to exhibit both bilateral symmetry and a triploblastic body plan. They are, in a sense, the first true movers and shakers of the animal kingdom, charting a new course for how life could organize itself. Despite their groundbreaking innovations, flatworms retain a certain elegant simplicity. Their most defining characteristic, as their name suggests, is their remarkably flattened body, which is dorsoventrally compressed like a ribbon or a leaf. This flatness isn't merely an aesthetic choice, it's a brilliant adaptation. Lacking specialized circulatory or respiratory systems, their flattened shape maximizes the surface area to volume ratio, allowing for efficient diffusion of gases and nutrients directly across their body surface. It's a testament to nature's ability to solve complex problems with elegant, often simple solutions. Let's delve a little deeper into the internal world of these fascinating pioneers. While they are triploblastic, flatworms are still acoelomate, meaning they lack a true fluid-filled body cavity, a column between their digestive tract and outer body wall. Instead, the space between their organs is filled with a solid mass of mesodermal tissue called parenchyma. This is a crucial distinction and a stepping stone to even greater complexity we will explore later. Their digestive system, while still incomplete, meaning it has only one opening that serves as both mouth and anus, is often highly branched, especially in free-living forms. This branching allows nutrients to be distributed more effectively throughout their flattened bodies, compensating for the lack of a circulatory system. Imagine a tiny tree of digestion, its branches reaching every corner. One of the truly significant evolutionary innovations we observe in flatworms is the emergence of a more organized excretory system. For the first time, we encounter protonephridia, often referred to as flame cells. These specialized cells, equipped with a tuft of cilia that beat rhythmically, resembling a flickering flame under the microscope, create a current that drives excess water and metabolic wastes through a network of tubules and out of the body through pores. This system is primarily responsible for osmoregulation, maintaining water balance, and is a far cry from the simple diffusion of earlier forms. It's a testament to the increasing demands of a more complex, active lifestyle. Their nervous system, too, shows remarkable progress compared to the diffuse nerve net of cnidarians. Flatworms possess a more centralized, ladder-like nervous system, with two anterior ganglia, clusters of nerve cells that act as a primitive brain, and two ventral nerve cords running the length of the body, connected by transverse commissures. This organization allows for more coordinated movement and a greater capacity for processing sensory information, and indeed, in many free-living flatworms, we find specialized sensory structures, chemoreceptors to detect chemicals in the water, and often a pair of light-sensitive eye spots or ochelli at their anterior end. While these eye spots can only detect the presence or absence of light, rather than forming images, they are a profound step towards true vision and further emphasize the advantages of cephalization. Reproduction in flatworms is often complex and varied. Many species are hermaphroditic, possessing both male and female reproductive organs, and engage in fascinating mating rituals, sometimes involving penis fencing, in which two individuals attempt to inseminate each other. Perhaps even more astonishing is the incredible regenerative capacity of many flatworms, particularly the free-living planarians. If you were to cut a planarian into several pieces, each piece, provided it's large enough, can often regenerate into a complete new individual. It's a superpower that continues to captivate scientists and hints at the remarkable plasticity of their developmental processes. Within the phylum Platyhelminthes, we find three major classes, each illustrating different facets of this foundational body plan. First, the Turbellaria, which includes the free-living flatworms, like our familiar planarians. These creatures are often found gliding gracefully over rocks and vegetation in freshwater ponds, marine environments, and even moist terrestrial habitats. They are typically predators or scavengers, using their keen chemoreceptors to locate food and their muscular pharynx to engulf it. Their vibrant colours and intricate patterns, coupled with their fluid movement, make them captivating subjects for quiet observation. They represent the ancestral free-living form, demonstrating the potential of bilateral symmetry and triploblasty without the constraints of a parasitic lifestyle. Then we encounter the Trematodagi, or flukes. These are fascinating examples of evolutionary specialization, as almost all flukes are internal parasites, endoparasites of vertebrates, with complex life cycles that often involve multiple hosts. Consider, for instance, the blood flukes of the genus Schistosoma, which affect hundreds of millions of people worldwide, causing debilitating diseases. Flukes possess suckers for attachment to their host's tissues and a tough outer cuticle that protects them from the host's digestive enzymes and immune responses. Their lives are a testament to the intricate dance of coevolution, where parasite and host have shaped each other's destinies over eons. Finally, we arrive at the Cestoda, the tapeworms. These are perhaps the most specialized and highly adapted of the flatworms, living exclusively as endoparasites in the digestive tracts of vertebrates. Tapeworms are truly remarkable in their adaptations. They completely lack a digestive system of their own. Instead, they absorb digested nutrients directly from their host's intestine across their body surface. Their bodies are composed of a small anterior region called the Golex, equipped with hooks and suckers for attachment, followed by a long chain of reproductive segments called proglottids. Each proglottid is essentially a self-contained reproductive factory, producing thousands of eggs. Tapeworms, like the beef or pork tapeworm, can grow to astonishing lengths, silently siphoning nutrients from their hosts, and their presence underscores the profound impact these seemingly simple creatures can have on the health of other animals, including ourselves. As we reflect on the flatworms, we see them as more than just a diverse group of creatures, some free-living and some parasitic. We see them as a crucial evolutionary stepping stone. They perfected bilateral symmetry and triploblasty, laying down the fundamental blueprint for almost all subsequent animal life. They introduced cephalization, a more centralized nervous system, and dedicated excretory structures. These innovations were monumental, opening up vast new possibilities for movement sensing and internal organization. But even with these advancements, the eco-element body plan presented certain limitations. The lack of a true body cavity meant that internal organs were tightly packed, restricting their independent movement and growth. It also limited the efficiency of circulation and waste removal in larger, more active bodies. And so, as we prepare for our next gentle exploration, we might find ourselves wondering, what would happen if that solid mesodermal packing could be replaced by a fluid-filled space? What new freedoms and complexities would a true body cavity, a coelom unlock. How would this allow for even greater development of organ systems, enabling animals to grow larger, move with more power, and develop even more intricate internal machinery? These are the questions that will guide our next quiet journey into the ever-unfolding story of animal life. For now, let us savour the profound elegance of the flatworms, these silent pioneers who first charted the path of bilateral symmetry, forever changing the course of evolution. As we gently drift away from the fascinating yet fundamentally simple architecture of the flatworms, we find ourselves on the cusp of another monumental evolutionary leap. We've seen how the development of bilateral symmetry and triploblasty in the platyhelminthes allowed for cephalization and the beginnings of directed movement, a crucial step away from radial symmetry. Yet, remember, these pioneering flatworms, for all their advancements, remained achoelomate. Their internal organs were packed within a solid mass of mesodermal tissue, a design that, while functional, presented certain limitations. Imagine for a moment an intricate machine where every component is tightly wedged against its neighbour, leaving no room for expansion, independent movement, or the cushioning of vital parts. This, in a simplified sense, was the challenge inherent in the achoelomate body plan. What if instead there were a fluid-filled space, a kind of internal buffer, that could offer support, flexibility, and a dedicated environment for organs to develop and function with greater autonomy? This, dear friends, is precisely where our journey takes us next, into the profound significance of a body cavity. Have you ever paused to truly consider the marvel of your own internal spaces? The way your heart beats rhythmically within its protective pericardial cavity, or your lungs expand and contract within the pleural cavities, all cushioned and supported, yet free to move. This intricate design, this ingenious solution to the challenges of internal organization, began to take shape eons ago with the emergence of true body cavities. Let us define our terms, as we always do in our tranquil corner of curiosity. A yesor bod cavity is, quite simply, an internal space within the body of an animal. But not all body cavities are created equal, and their evolutionary journey is a testament to life's persistent innovation. We can broadly categorize animals based on the presence and nature of their body cavity. Firstly, the achoelomates, like our flatworm friends, meaning without a coelom. As we've discussed, they lack a fluid-filled body cavity between their digestive tract and outer body wall. The space is instead filled with mesodermal parenchyma. Next, we encounter the pseudocoelomates, meaning false coelom. These animals possess a body cavity, but it is not completely lined by mesoderm. Instead, the mesoderm lines the outer body wall, but the digestive tract remains suspended within the cavity without a mesodermal covering. Think of it as a partially finished room. The walls are there, but the inner furnishings aren't fully integrated with the structure. And finally, the eucoelomates, or true coelomates. Here, the body cavity, known as a coelome, is entirely lined by mesoderm, which forms a peritoneum that suspends and covers the internal organs. This is the most complex and evolutionarily advanced form of body cavity, offering the greatest advantages. Why is this distinction so vital? What profound advantages did the evolution of a body cavity, even a false one, confer upon early animals? Imagine the possibilities. 1. A hydrostatic skeleton. For soft-bodied animals, a fluid-filled cavity provides internal support against which muscles can contract. This allows for far more sophisticated and powerful movements than simply relying on muscle contractions against solid tissue. It's like having an internal water balloon that you can squeeze to change your shape and propel yourself forward. 2. Space for organ development and growth. A body cavity offers a dedicated area for organs to grow, differentiate, and mature without being constricted by surrounding tissues. This allows for greater complexity and specialization of organ systems. 3. Enhanced circulation and transport. The fluid within the body cavity can serve as a medium for transporting nutrients, gases, and wastes throughout the body, especially in smaller animals or those without a dedicated circulatory system. 4. Protection of internal organs. The fluid acts as a cushion, protecting delicate organs from external shocks and internal movements. 5. Increased body size potential. By providing internal support and space, a body cavity allows animals to achieve larger body sizes and more complex three-dimensional forms. 6. Independent movement of body wall and internal organs. With a coelom, the digestive tract and other internal organs can move somewhat independently of the outer body wall, facilitating processes like digestion and peristalsis without affecting external locomotion. With these remarkable benefits in mind, let us now turn our attention to the first major phyla that embraced the innovation of a body cavity, even if it was a pseudocoelom. We journey now into the realm of the phylum Nematoda, the ubiquitous, often unseen but profoundly important roundworms. Step with us into their world and you might find it astonishingly diverse and widespread. Nematodes are quite simply everywhere, from the deepest ocean trenches to the highest mountain peaks, from the soil beneath your feet to the internal tissues of nearly every plant and animal. These cylindrical creatures thrive. If you were to magically remove all matter from Earth except for nematodes, their ghostly forms would still outline the contours of continents and the bodies of living things. Such is their sheer abundance and ecological significance. What defines these prolific creatures? Firstly, like the flatworms, nematodes are bilaterally symmetrical and triploblastic, meaning they develop from 3 germ layers. However, they are distinguished by their distinct, unsegmented, cylindrical body plan, which typically tapers at both ends. This gives them their characteristic roundworm appearance. The most crucial evolutionary advancement we observe here is their pseudocoelom pore. This fluid-filled cavity provides the hydrostatic skeleton we just discussed, allowing nematodes to achieve their characteristic whip-like undulating movement. They possess only longitudinal muscles, running the length of their body. When these muscles contract, they can only shorten one side, causing the body to bend. Because there are no circular muscles, they cannot constrict their body diameter, leading to that distinctive thrashing motion. Another significant leap forward is the development of a complete digestive tract. Unlike the gastrovascular cavity of cnidarians and flatworms, which has a single opening serving as both mouth and anus, nematodes possess a distinct mouth at one end and an anus at the other. This tube-within-a-tube body plan allows for unidirectional food processing, meaning food moves in one direction, allowing for specialized regions for digestion and absorption along the length of the gut. This is a far more efficient system than the two-way traffic of a gastrovascular cavity. Their outer body is covered by a tough, flexible, non-cellular layer called a cuticle. This cuticle provides protection against desiccation, physical damage, and host defenses for parasitic forms. It must be molted periodically as the nematode grows, a process called ecdysis, which links them to the other ecdysozoans, a superphylum we will explore in due course. Nematodes exhibit a range of lifestyles. Many are so free-living, playing vital roles in soil ecology as decomposers and predators of bacteria, fungi, and other small invertebrates. Others are notorious for parasites, infecting plants, insects, and vertebrates, including humans. Think of the microscopic Caenorhabditis elegans, often simply called C. elegans, a free-living soil nematode that has become one of the most important model organisms in biological research, revolutionizing our understanding of genetics, development, and neuroscience. Then there are the more infamous parasitic forms, hookworms, which can cause anemia, Ascaris lumbricoides, large intestinal roundworms, pinworms, common in human children, and filarial worms, responsible for debilitating diseases like elephantiasis and river blindness. The sheer diversity of their parasitic strategies is a testament to their evolutionary success. As we ponder the ubiquitous presence and ecological importance of nematodes, let us also consider another fascinating group of pseudo-coelemates, often overlooked due to their microscopic size, but no less remarkable: the phylum Rotifera, commonly known as rotifers or wheel animalcules. To observe a rotifer under a microscope is to witness a tiny pulsating marvel of life. These predominantly freshwater inhabitants are truly a spectacle, often resembling delicate, transparent jewels. Like nematodes, they are bilaterally symmetrical and triploblastic, possessing a pseudo-coelom. Their most distinctive feature and the source of their charming name is the corona, Latin for crown, a ciliated organ located at their anterior head end. The rhythmic beating of these cilia creates a swirling current that resembles rotating wheels, hence wheel animalcules. This corona serves a dual purpose. It generates a current to draw food particles like bacteria, algae, and detritus into their mouth, and it also aids in locomotion, propelling them through the water. Rotifers too possess a complete digestive tract, a testament to the efficiency of this body plan. Within their pharynx, they house a unique, complex and muscular structure called the mastax, which contains hard, chitinous jaws called ultrophy. These trophy are used for grasping, grinding and crushing food particles, a specialized feeding apparatus that allows them to process a wide range of microscopic fare. Their reproductive strategies are also quite captivating. Many species exhibit parthenogenesis, a form of asexual reproduction where females produce offspring from unfertilized eggs. Some species are entirely parthenogenetic, with males being rare or even unknown. This ability allows for rapid population growth in favorable conditions. Despite their minute size, rotifers are critical components of freshwater ecosystems. They are significant primary consumers, grazing on algae and bacteria, and in turn serve as an important food source for larger invertebrates and fish larvae. They are truly the unsung heroes of the aquatic microbial world, silently maintaining the balance of life in ponds and lakes. So as we conclude this chapter, we reflect on the journey from the solid acoelemate body plan of the flatworms to the emergence of the fluid-filled pseudocoelom in the nematodes and rotifers. This seemingly simple internal space has unlocked a cascade of evolutionary possibilities: more efficient movement, enhanced organ development, and sophisticated feeding strategies. It is a powerful reminder that sometimes the greatest innovations lie in creating space for growth, space for function, space for life to flourish in ever more complex and astonishing ways. Next, we will venture into the realm of the true coelom, where the mesoderm fully embraces and lines the internal organs, paving the way for even greater anatomical sophistication and the incredible diversity of life we are yet to explore. Join us as we continue our quiet explorations into the bedrock of biology. As we gently drift from the fascinating world of the pseudocoelemates, those remarkable roundworms and rotifers with their partially lined body cavities, we find ourselves on the cusp of an evolutionary epoch. Have you ever paused to truly consider what a profound difference a subtle anatomical shift can make? Last time we touched upon the concept of the true coelom, that fully mesoderm-lined internal space that offers such a wealth of biological advantages. Today we invite you to step with us across this pivotal threshold, to explore the true coelom in all its glory, and to witness the grand evolutionary divergence it ushered in, the split between protostomes and deuterostomes. Imagine for a moment a magnificent, intricate machine. Its gears turn, its levers pull, its systems hum in perfect synchronicity. Now picture that machine without any internal cushioning, without separate compartments for its vital components, or with parts that cannot move independently. It would be a far less efficient, far more fragile creation, wouldn't it? This analogy, perhaps, offers a glimpse into the profound leap from an acoelemate, or even a pseudo-coelemate body plan to one graced with a true coelom. What precisely distinguishes this true cavity? We recall that in pseudo-coelemates, the body cavity, though present, is only partially lined by mesoderm, the middle germ layer. It's a space, yes, but one that feels a little less integrated, a little less finished in its design. The true coelom, in contrast, is entirely enveloped by mesodermal tissue. This means that the outer body wall and the internal organs are both suspended within this cavity by mesodermal membranes. It's not just a space, it's an organized space, a fully developed internal architecture that allows for unprecedented levels of complexity and functional sophistication. This complete mesodermal lining bestows a series of invaluable gifts upon the organisms that possess it. First, and perhaps most intuitive, is the enhanced hydrostatic skeleton. While pseudo-coelemates could utilize their internal fluid for support and movement, the true coelom, with its fully enclosed and often more voluminous fluid-filled space, allows for much finer, more controlled movements. The mesodermal lining often gives rise to muscles that can act antagonistically against the coelomic fluid, enabling precise changes in body shape and locomotion. Think of an earthworm, a classic eucoelemate. Its segmented body, each segment with its own fluid-filled coelom, allows for sophisticated peristaltic movement, pushing and pulling against the ground with remarkable efficiency. Without that true coelom, such coordinated locomotion would be a distant dream. Secondly, the true coelom provides ample space for the development and specialization of complex internal organs. No longer are organs packed tightly against the body wall or each other. They can grow, expand, and differentiate into highly specialized structures, each performing its unique function without impeding its neighbors. This generous internal volume allows for the evolution of more elaborate digestive systems, circulatory systems, excretory organs, and reproductive structures. It's like moving from a cramped studio apartment to a spacious mansion, where every room can be designed for a specific purpose, optimizing comfort and function. Furthermore, this mesodermal suspension means that the internal organs can move independently of the outer body wall. Imagine a world where your stomach couldn't churn without your entire torso twisting, or your heart couldn't beat without your skin rippling. This is, to some extent, the reality for animals without a true coelom. But with a true coelom, the gut can process food, the heart can pump blood, and the reproductive organs can mature, all while the animal is moving, burrowing, or simply resting. This independence allows for greater physiological efficiency and behavioral flexibility, enabling animals to perform multiple vital functions simultaneously. The coelom also plays a critical role in circulation and transport. In many eucoelemates, the coelomic fluid itself can act as a circulatory medium, transporting nutrients, gases, and waste products throughout the body. In more complex forms, it provides the space for the development of sophisticated closed circulatory systems, ensuring efficient delivery of resources to every cell. It's also an excellent internal buffer, cushioning organs against external impacts and providing a stable internal environment, much like the shock absorbers in a finely tuned vehicle. With these profound advantages, the true coelom became a cornerstone of animal evolution, opening up vast new possibilities for diversification and complexity. Indeed, the emergence of the true coelom marks one of the most significant evolutionary innovations in the history of life, setting the stage for the incredible array of animals we know today. Now, as we delve deeper into the eucoelemates, we encounter another monumental branching point, a fundamental divergence in the very earliest stages of embryonic development that divides the vast majority of true coelemates into two grand lineages, the protostomes and the deuterostomes. This distinction is not merely academic, it reflects deeply ingrained differences in how these animals construct their bodies from a single fertilized egg. It's a testament to the fact that even subtle variations in developmental pathways can lead to entirely different evolutionary trajectories. Let us first turn our attention to the protostomes. The name itself, derived from Greek, means mouth first, hinting at a key developmental characteristic. In protostomes, the blastopore, the first indentation that forms during gastrulation, the process where the single-layered blastula reorganizes into a multi-layered structure, develops into the animal's mouth. The anus, if present, forms later, at the opposite end of the embryo. Beyond this, protostomes exhibit several other defining embryonic features. Their cleavage, the series of rapid cell divisions after fertilization, is typically spiral cleavage. Imagine the cells dividing and stacking not directly on top of each other, but at an angle, creating a spiral arrangement of cells. This contrasts sharply with the radial cleavage we will soon discuss. Furthermore, protostomes generally display determinate cleavage. This means that the developmental fate of each embryonic cell is fixed very early on. If you were to remove a single cell from a protostome embryo at an early stage, the remaining cells would develop into an incomplete organism, and the removed cell would develop into only a partial embryo. Each cell's destiny is predetermined, a specific piece of the overall developmental puzzle. The formation of the coelom itself in protostomes is also distinctive, a process called schizocolous coelom formation. As the mesoderm germ layer begins to form, it does so as solid blocks of tissue that then split apart or schiz to create the coelomic cavity. Think of carving a space out of a solid block rather than forming it from an outpouching. The protostome lineage is incredibly diverse and successful, encompassing a vast array of familiar animals. Within this group we find two major clades that you will come to know well, the Lophotrochozoa and the Ausa ecdysozoa. The Lophotrochozoa include animals characterized either by a feeding structure called a lophophore, a ciliated crown of tentacles, or by a specific larval type known as a trochophore larva. This diverse group includes the segmented worms, phylum Annelida, the mollusks, phylum Mollusca, with their incredible variety of forms from snails to octopuses and several other fascinating phyla. The ecdysozoa, on the other hand, are united by the shared characteristic of ecdysis, or molting, the periodic shedding of a tough external cuticle or exoskeleton to allow for growth. This group includes the incredibly numerous and diverse arthropods, phylum Arthropoda, insects, spiders, crustaceans, which are arguably the most successful animal phylum on Earth, as well as the nematodes, phylum Nematoda, we met in our last chapter. Even though they are pseudo-coelamates, their molting places them firmly within this clade. The sheer variety and ecological dominance of protostomes speak volumes about the evolutionary success of their developmental blueprint. On the other side of this grand developmental divide lie the deuterostomes. Their name, again from Greek, means mouth second, revealing their contrasting developmental path. In deuterostomes, the blastopore, that initial embryonic indentation, develops into the anus. The mouth forms secondarily, at a later stage of development, typically at the opposite end of the embryo. This simple reversal of mouth and anus formation is a fundamental difference with profound evolutionary implications. Deuterostomes also exhibit distinct patterns of cleavage. Their cleavage is radial cleavage, meaning the cells divide and stack directly on top of each other, forming neat aligned rows of cells. This creates a symmetrical radial arrangement, and unlike the determinate cleavage of protostomes, deuterostomes typically display indeterminate cleavage. This means that the developmental fate of each embryonic cell is not fixed early on. If you were to separate a cell from an early deuterostome embryo, each isolated cell has the potential to develop into a complete, albeit smaller organism. This characteristic is why identical twins are possible in humans and other deuterostomes. The early embryonic cells retain their totipotency, their abilities develop into any cell type. The formation of the column in deuterostomes is also different, known as enterocoelous coelom formation. Here the column forms from outpocketings of the archenteron, the primitive gut. Imagine the gut wall pushing outwards, forming pouches that eventually pinch off to become the coelomic cavity. It's a process of budding and expansion rather than splitting. The deuterostome lineage, while perhaps less numerically diverse than the protostomes, includes some of the most complex and familiar animals, including ourselves. This group comprises the echinoderms, phylum Echinodermata, those fascinating marine animals like sea stars and sea urchins, and, most notably for us, the chordates, phylum Chordata, which include all vertebrates, fish, amphibians, reptiles, birds, and mammals. As we conclude this exploration of the true coelom and the ancient split between protostomes and deuterostomes, we hope you feel the same sense of profound wonder that we do. These seemingly minute differences in early embryonic development laid the foundation for the two major evolutionary highways that animal life would travel, leading to the astonishing diversity we see around us. The true coelom, in all its fully lined glory, provided the internal scaffolding and flexibility for organ to specialize, for movement to become precise, and for bodies to grow complex. And the protostome-deuterostome divide established fundamentally different blueprints for constructing these complex bodies. In our next quiet explorations, we will begin to journey down these highways, starting with the protostomes to meet the incredible array of creatures that exemplify these developmental patterns. We will unravel the intricate anatomies of segmented worms, the ingenious designs of mollusks, and the extraordinary adaptations of arthropods. For now, let this tranquil corner of curiosity allow you to contemplate the elegant simplicity of these foundational principles and the boundless potential they unlocked in the grand narrative of life. As we gently drifted from our quiet explorations of the pseudo-coelamates, we arrived at a truly profound divergence in the grand narrative of animal life, the split between more protostomes and deuterostomes. This was not merely a branching of a few species, but a fundamental fork in the evolutionary road, defining two vast and incredibly successful lineages. Remember, we spoke of the true coelom, that mesoderm-lined cavity offering such remarkable advantages for organ development and independent movement. Now we shall truly appreciate how this innovation set the stage for two distinct pathways of embryonic development, each leading to an astonishing array of forms. Today, in our tranquil corner of curiosity, we turn our patient contemplation towards the protostomes. Have you ever paused to truly consider how the very first moments of an embryo's life can dictate the entire architectural plan of an animal? For protostomes, it's a story of mouth first. The term itself, proto meaning first and stoma meaning mouth, whispers its primary defining characteristic, the blastopore. That initial indentation in the developing embryo becomes the mouth. But the story of protostome development is richer than this single detail. Imagine, for a moment, the earliest cellular divisions, like tiny specialized workers beginning to lay the foundations of a grand, living city. In protostomines, these early cell divisions follow a spiral cleavage in pattern, where new cells are offset from the parent cells, creating a beautiful spiraling arrangement. And this cleavage is often more determinate, meaning the fate of each embryonic cell is largely set early on. If you were to remove one of these early cells, the developing embryo would likely be incomplete, a testament to the pre-programmed nature of its design. Then comes the formation of the coelom itself, that ingenious internal space. In protostomes, the coelom forms through a process called schizocoelous development. Picture the mesoderm, that vital middle germ layer, splitting to create a cavity. It's as if the walls of a room are being pushed apart from within, creating a new vital space. This combination of mouth-first development, spiral and determinate cleavage, and schizocoelous coelom formation defines the vast and varied world of the protostomes, a world we are about to explore. Within this expansive protostome lineage, zoologists have identified two major clades, or superphyla, that further organize the incredible diversity we observe. These are the Lophotrochozoa and the Ecdysozoa. Each represents a distinct evolutionary strategy, a different set of adaptations that have allowed them to flourish across nearly every conceivable habitat on Earth. Let us first gently drift into the realm of the Lophotrochozoa. This clade is named for two distinct larval forms or feeding structures found in many of its members. Some possess a lophophore, a magnificent crown of ciliated tentacles surrounding the mouth, used for suspension feeding. Imagine a delicate living filter, sweeping microscopic particles from the water, a truly elegant solution for sustenance. Others, though not all, share a characteristic larval stage known as a trochophore larva, a free-swimming, top-shaped larva adorned with bands of cilia. These tiny ciliated wanderers are a testament to shared ancestry, a whisper from deep time connecting seemingly disparate groups. Among the Lophotrochozoa, two phyla stand out for their widespread presence and evolutionary significance: the Annelida, the segmented worms, and the mollusca, a phylum of astonishing diversity encompassing everything from snails to octopuses. Consider first the phylum Annelida, the segmented worms. When you think of an earthworm, perhaps after a gentle rain, you're observing a masterwork of biological engineering. Their most striking feature, as their name suggests, from the Latin annulus, meaning little ring, is their segmentation. Their bodies are composed of a series of repeating units or segments, each often containing duplicate sets of organs and muscles. Isn't it remarkable how this modular design offers such resilience and specialization? If one segment is damaged, others can often continue to function, and specialized segments can evolve for specific tasks like reproduction or sensation. This segmentation extends to their true coelom, which is also segmented, creating a series of fluid-filled compartments. This compartmentalized coelom acts as a powerful hydrostatic skeleton, allowing annelids to perform complex movements like burrowing. By contracting muscles in different segments, they can lengthen, shorten, and expand individual parts of their body, pushing through soil or swimming gracefully. Annelids boast a closed circulatory system, meaning their blood is always contained within vessels, much like our own. This allows for efficient transport of nutrients, gases, and wastes throughout their elongated bodies. Their nervous system is equally sophisticated, featuring a brain-like pair of ganglia in the head region and a ventral nerve cord running the length of their body, with smaller ganglia in each segment coordinating localized activities. From the familiar earthworms, oligochaetes, that aerate our soil, enriching it with their tireless work, to the vibrant, often ornate marine polychaetes with their feathery gills and fierce jaws, and even the medicinal leeches, annelids demonstrate an incredible range of adaptations. They are silent alchemists of the soil, vital components of marine ecosystems, and fascinating examples of the power of segmentation. Now, let us turn our attention to the phylum Mollusca, a group that truly embodies the phrase astonishing diversity. From the slow, gentle grace of a garden snail to the intelligent, quicksilver movements of an octopus, mollusks captivate us with their varied forms and behaviors. Yet beneath this outward variety lies a remarkably consistent underlying body plan, a testament to shared ancestry. Most mollusks possess three main body parts: a muscular foot, used for locomotion or attachment, a visceral mass, which contains most of the internal organs, digestive, excretory, reproductive, and a mantle, a fold of tissue that drapes over the visceral mass. This mantle often secretes a protective shell, though in some mollusks like squids and octopuses, the shell is reduced or entirely absent. A unique feature found in most mollusks is the radula, a chitinous ribbon-like structure armed with rows of tiny teeth, used for scraping food from surfaces or even drilling into other shells. Imagine a microscopic conveyor belt of teeth, constantly renewed, allowing for a wide range of feeding strategies. Within the mollusca, we encounter several well-known classes. The gastropods, meaning stomach foot, include snails and slugs. They glide on their muscular foot, often protected by a coiled shell, and graze on algae or plants. Have you ever paused to truly consider the intricate beauty of a snail's shell? A marvel of natural geometry. Then there are the bivalves, such as clams, oysters, and mussels. Their bodies are compressed between two hinged shells, hence bivalve. They are typically filter feeders, drawing water into their mantle cavity and straining out food particles. These quiet, sedentary creatures play crucial roles in purifying aquatic environments. And finally, the utterly captivating cephalopods, meaning headfoot, which include octopuses, squids, cuttlefish, and nautiluses. These are the most active and intelligent of the mollusks, with highly developed nervous systems, complex eyes, and a foot modified into tentacles and a siphon for jet propulsion. Their ability to change colour and texture in an instant for camouflage or communication is nothing short of mesmerising. They are truly the apex predators of the invertebrate world, a testament to the evolutionary potential of the mollusk body plan. Having savoured the wonders of the Lophotrochozoa, we now pivot to the second great protostome clade, the Ecdysozoa. This group is united not by a larval form or feeding structure, but by a shared physiological process, ecdysis, or molting. All ecdysozoans grow by shedding an external cuticle or exoskeleton. Imagine a knight shedding an old too small suit of armour to grow into a new, larger one. This periodic shedding is a vulnerable but necessary part of their life cycle, allowing for growth and sometimes dramatic transformations. The undisputed champions of the Ecdysozoa, and indeed of the entire animal kingdom in terms of species diversity and sheer numbers, are the members of the phylum Arthropoda. The name Arthropoda itself, meaning jointed feet, hints at their most defining characteristics: a tough-gunned exoskeleton, a segmented body, and highly specialized jointed appendages. The exoskeleton, typically made of chitin, provides crucial protection against predators and desiccation and offers attachment points for muscles, enabling powerful movements. However, as we discussed, it also necessitates molting, a period of vulnerability. Their segmented bodies are often organized into distinct regions or tagmata, such as a head, thorax, and abdomen, each specialized for different functions like feeding, locomotion, or reproduction. Their jointed appendages, whether legs, antennae, or mouthparts, are incredibly versatile, having evolved for a bewildering array of tasks. The success of arthropods is truly unparalleled. They dominate nearly every terrestrial, freshwater, and marine ecosystem. We can broadly categorize them into several major subphyla. Chelicerates, including spiders, scorpions, ticks, and mites, characterized by claw-like feeding appendages called chelicerae. Myriapods, such as centipedes and millipedes, known for their elongated bodies with many pairs of legs. Crustaceans, a largely aquatic group including crabs, lobsters, shrimp, and barnacles, often possessing 2 pairs of antennae. And finally, the most diverse group of all, the hexapods, which primarily consist of insects. Insects, with their incredible adaptations for flight, complex life cycles often involving metamorphosis, from larva to pupa to adult, and highly specialized feeding strategies, represent an evolutionary pinnacle of the arthropod body plan. From the silent, intricate dance of a spider spinning its web, to the industrious hum of a bee pollinating flowers, to the powerful migrations of monarch butterflies, arthropods are constant reminders of life's boundless capacity for adaptation and innovation. Their sheer abundance and ecological impact are staggering, shaping the very fabric of ecosystems worldwide. As we conclude our initial journey into the captivating world of the protostomes, we hope you feel a deeper appreciation for the foundational, evolutionary choices made billions of years ago. The mouth-first development, the spiral cleavage, the schizocolous coelom formation, these seemingly small embryological details have unfolded into the magnificent diversity we see in the segmented worms, the intelligent octopuses, and the ubiquitous insects. We have merely scratched the surface of these remarkable lineages, each phylum a universe unto itself, brimming with unique adaptations and life stories. But for now, we leave the bustling, molting, and spiraling world of the protostomes, carrying with us a renewed sense of wonder for their ingenuity and their enduring success. In our next quiet exploration, we shall turn our gaze to the other great branch of eucoelomate evolution, the deuterostomes, and discover how their anus-first developmental pathway has led to yet another astonishing tapestry of life, including our own. And so we arrive gently at another monumental crossroads in our shared exploration of the animal kingdom, don't we? Having journeyed through the intricate and astonishing world of the protostomes, those incredible architects of diversity, from the segmented marvels of the annelids and the soft-bodied ingenuity of the mollusks to the armoured legions of the arthropods, we now find ourselves poised at the threshold of their evolutionary counterparts. Imagine, for a moment, standing at a grand divergence, a point where the river of life split into two mighty currents, each destined to carve its own magnificent path through the landscape of deep time. We have followed one, and now, with quiet anticipation, we turn our gaze to the other. Welcome, dear friends, to the realm of the deuterostome. If you recall, our previous contemplations illuminated the fundamental differences that set these two superphyla apart, differences etched into the very earliest moments of their embryonic development. While protostomes famously declare mouth first in their construction, the deuterostomes, with a quiet elegance, whisper anus first. It is a distinction at first glance that might seem merely a curious detail, yet it is a profound divergence that has shaped the entire trajectory of life, leading to forms as disparate as the spiny inhabitants of the ocean floor and, indeed, to us. Let us briefly revisit those foundational embryonic distinctions, shall we? For the deuterostomes we observe radial cleavage, or where the planes of cell division are either parallel or perpendicular to the polar axis of the embryo, resulting in tiers of cells directly above or below each other. This is wonderfully contrasted with the spiral cleavage we found in many protostomes. And then there is indeterminate cleavage, a truly remarkable feature. Here, each early embryonic cell retains the potential to develop into a complete organism should it become separated from its siblings. This is why identical twins are possible in humans, for instance, a testament to our deuterostome heritage. This stands in stark contrast to the determinate cleavage of protostomes, where the developmental fate of each cell is set very early on. Finally, the formation of the coelom itself, that crucial body cavity we've come to appreciate so deeply, also follows a different script. In deuterostomes, the coelom forms via enterocolous development, where folds of the archenteron, the primitive gut, pinch off to form the colon. This is a wonderfully elegant process, distinct from the schizocolous formation we observed in many protostomes, where the colon simply splits within the mesoderm. These are not mere academic distinctions, you see. They are the silent, foundational blueprints that dictate the very possibilities of form and function. They are the whispers of life's earliest decisions, reverberating through countless millennia. Now, as we step into this new evolutionary landscape, we encounter a relatively smaller yet no less significant collection of phyla. The deuterostomes include some of the most iconic and, to us, most intimately familiar groups of animals. But before we arrive at the grand lineage that ultimately led to the vertebrates, to us with our complex brains and articulated skeletons, we must first pause and marvel at our closest invertebrate cousins, a group of truly unique and ancient marine creatures, the phylum Echinodermata. Have you ever paused to truly consider the sea star, perhaps glimpsed one clinging to a rock in a tide pool, or seen the intricate patterns of a sand dollar? These are the echinoderms, a name that in its gentle translation means spiny skin, a characteristic that immediately hints at their distinctive appearance. This phylum encompasses the sea stars, often still affectionately called starfish, though they are not fish at all, brittle stars, sea urchins, sand dollars, and sea cucumbers, alongside the more ancient and often sessile crinoids, or sea lilies. All are exclusively marine, a testament to their deep evolutionary roots in the ocean's embrace. What strikes us first about an adult echinoderm is often its unique symmetry. While their larvae are unequivocally bilaterally symmetrical, a clear echo of their ancestral heritage, the adults undergo a fascinating metamorphosis, adopting a secondary pentaradial symmetry. This means their body parts are arranged in multiples of 5 around a central axis. Imagine a sea star with its 5 arms radiating outwards, or a sea urchin whose spherical body is subtly organized into 5 distinct sections. This shift from bilateral to radial symmetry in adulthood is a profound evolutionary adaptation, one that often correlates with a more sessile or slow-moving existence, allowing them to perceive their environment from all directions equally. It's as if they have chosen to become living mandalas of the ocean floor. Beneath that often spiny, often leathery skin lies another defining feature, an endoskeleton, composed of calcareous plates or ossicles. These plates, derived from the mesoderm, are embedded within the dermis and can be fused together, as in the rigid test of a sea urchin, or loosely articulated, providing flexibility, as in the arms of a sea star. This internal skeleton offers both support and protection, a different evolutionary strategy from the external exoskeletons we so admired in the arthropods. It is a living armour, growing with the animal, often adorned with spines that can be fixed or movable, adding another layer of defence against predators. But perhaps the most truly astonishing and unique characteristic of the echinoderms, the very heart of their physiological marvel, is their water vascular system. This intricate network of fluid-filled canals, reservoirs and tube feet is unlike anything else in the animal kingdom, a true testament to nature's boundless creativity. Let us trace its path. Seawater enters the system through a sieve-like plate on the arboreal upper surface called the madreporite. From there, it flows down a short stone canal to a circular ring canal encircling the central disc. Radiating outwards from the ring canal are radial canals, extending into each arm or body section. Attached to these radial canals are hundreds, sometimes thousands, of small muscular sacs called ampullae, each connected to a slender external projection known as a tube foot. Imagine these ampullae as tiny squeezable bulbs. When an ampulla contracts, it forces water into the attached tube foot, extending it. The tube foot then presses against a surface and its tip, often equipped with a suction cup, adheres. When the ampulla relaxes, water is withdrawn and the tube foot shortens, releasing its grip. By coordinating the extension and retraction of thousands of these tube feet, echinoderms achieve a remarkable, slow yet incredibly powerful form of locomotion. It is a silent hydraulic ballet, allowing them to glide across the seabed, cling to rocks, or even pry open the shells of bivalve prey. Beyond movement, this ingenious system also plays crucial roles in feeding, gas exchange, and waste excretion, making it a multifunctional marvel of biological engineering. Given their radial symmetry as adults, it is perhaps unsurprising that most echinoderms lack true cephalization. There is no distinct head, no concentrated brain at one end. Instead, their nervous system typically consists of a nerve ring around the mouth and radial nerves extending into each arm, allowing for coordination of their tube feet and sensory input from all directions. It is a decentralized elegance, perfectly suited to their lifestyle. And speaking of remarkable abilities, many echinoderms possess an extraordinary capacity for regeneration. A sea star, for instance, can often regrow a lost arm, and in some species, a single arm, provided it retains a portion of the central disc, can regenerate an entire new individual. This speaks to a profound resilience, a quiet determination to persist in the face of adversity, a biological testament to their ancient lineage. Let us briefly consider the diverse forms within this fascinating phylum. The asteroidea, or sea stars, are perhaps the most recognizable. With their typically 5 arms radiating from a central disc, they are often active predators, especially of bivalves. They employ their powerful tube feet to pry open shells, then, in a truly astonishing feat, avert their stomach through their mouth into the bivalve's shell, digesting the prey externally before retracting their stomach. A rather dramatic dining experience, wouldn't you agree? Then we have the Ophiuroidea, the brittle stars. Similar in general appearance to sea stars, their arms are typically more slender and distinctly set off from the central disc, and they move with a much more active, snake-like wriggling motion of their arms. They are often scavengers or suspension feeders, quietly sifting through the detritus of the ocean floor. The Echinoidea include the sea urchins and sand dollars. Sea urchins are spherical, often covered in long, movable spines that serve both for locomotion and defense. They are typically herbivores, grazing on algae with a unique and complex chewing apparatus called Aristotle's lantern, a marvel of biological mechanics. Sand dollars, on the other hand, are flattened, disc-shaped urchins, perfectly adapted for burrowing into sandy substrates. The Holothuroidea, or sea cucumbers, present a wonderfully different body plan. Their bodies are elongated along the orolateral axis, giving them a leathery, worm-like appearance, though their 5 rows of tube feet still betray their echinoderm identity. They are primarily deposit feeders, sifting organic matter from the seabed. When threatened, some species exhibit a truly spectacular defense mechanism. They can eviscerate, expelling their internal organs, which can then be regenerated later. A bold sacrifice indeed. Finally, the Crinoidea, or sea lilies and feather stars, represent some of the most ancient echinoderms. Sea lilies are sessile, attaching to the seabed by a stalk, while feather stars are motile. Both are suspension feeders, using their feathery branched arms to filter plankton from the water column. They are living relics, offering us a glimpse into the deep past of these remarkable creatures. Echinoderms play vital ecological roles in marine ecosystems. As grazers, predators, and detritivores, they help to structure benthic communities and maintain the health of coral reefs and seagrass beds. Their presence, often subtle, is a key thread in the rich tapestry of oceanic life. As we conclude our contemplation of the echinoderms, we find ourselves at the very doorstep of another, even more familiar lineage within the deuterostomes. These spiny-skinned marvels, with their unique symmetry and hydraulic systems, are surprisingly our closest invertebrate relatives. They are a testament to the diverse paths evolution can take, even from a shared ancestral blueprint. Next, dear friends, we will turn our attention to the lineage that, through a series of profound innovations, would eventually give rise to the very creatures that possess a backbone, to the vertebrates, and ultimately to us. It is a journey from the simple yet elegant to the complex and self-aware, a story that continues to unfold with every beat of a heart, with every thought that stirs within us. Until then, I invite you to carry the wonder of the echinoderms with you, a quiet reminder of the ocean's enduring magic and the deep shared history of all life.